In this experiment, sepia brown-eyed winged female flies were crossed with males that had red eyes and no wings. In the F1 offspring, there were 10 sepia males and 8 sepia females. One male had red eyes and no wings. 5 of these F1 sepia males were then crossed with 5 of the F1 sepia females. The F2 offspring consisted of 8 sepia males and 22 sepia females. It was determined that the sepia traits, those with brown eyes and wings, were dominant over the wingless and red-eyed traits. And here we have a wingless fly, and here we have one with wings. Genetics is one of the most interesting fields in science because it tells us why we are the way we are. It also tells us what our children might look like, and more importantly, if we will pass certain diseases on to them. Using genetics, we can predict traits like eye color, hair color, and height in our own children. A lot of the lessons we learn about genetics come from studying model organisms like fruit flies. The scientific name for the common fruit fly is Drosophila melanogaster. Other commonly used model organisms include mice, rats, worms, sea urchins, frogs, zebrafish, and certain kinds of plants, which are shown here. And here we have our fruit fly. These make great model organisms because they are able to produce large amounts of offspring in short periods of time. They have short life cycles, and they can survive in a laboratory environment fairly cheaply. Some of the earliest work on fruit flies was done by a man named Thomas Hunt Morgan in the early 1900s. Here he is. He looked at eye color and body color in the flies. Morgan discovered a mutant white-eyed fly, which was this guy here. But the most interesting thing was that all the flies with white eyes were males. This was the first time someone had uncovered a sex-linked trait. In humans, one sex-linked trait many of us are familiar with is red-green color blindness. Because the trait is carried on the X chromosome, mostly males end up having the disorder. Males only have one X chromosome, while females have 2. This means that females have a better chance of getting a healthy X chromosome that prevents them from having red-green color blindness. In the present study, the traits being looked at were the presence or absence of wings and eye colors in the flies. 7 red-eyed wingless males were crossed with 6 sepia females. It was hypothesized that the sepia traits, the wings and brown eyes, would be dominant to the red eyes and no wings. The first method used involved sexing the fruit flies. It had to be determined which ones were males and which were females, and their phenotypes had to be recorded. In order to determine the sex of the flies, we looked at the posterior abdomen, which is this bottom part right here. In males, the end of the abdomen is darker and more blunt. In females, the end of the abdomen is pointier and lighter in color. See the difference? The males also have sex combs on their forelegs, as pictured here. So it's a dark spot on their legs right here, and it looks like a little bit of a comb if you look really close up. In general, males are also smaller than the females, but that's not necessarily a reliable factor. Next, we had to make the medium for the vials. In the vials, we placed 1 scoop of instant Drosophila medium, found here, 1 scoop of tap water, and 4 to 6 grains of yeast. No more, no less. 7 A males with red eyes and no wings were placed in the vial, along with 6 C females with wings and brown eyes. A sponge was stuck in the top of the vial to keep the flies from escaping. These are what the vials looked like when they were all finished. After 7 days, it was time to clear the vials. To clear the parent generation out of the vials, a wand with fly nap was stuck into the vial against the sponge, as seen here. Once the flies had all gone to sleep, they were dumped from the vials and placed in the fly morgue, which was a petri dish containing ethanol. We used clean paint brushes to push some of the flies out. The F1 larvae remained in the vials, and we waited another 7 days for them to hatch. 7 days later, it was time to score the F1 flies. We sexed them and recorded their phenotypes the same way as before. We then set up the F2 vials with the same medium as before. 5 males with brown eyes and wings were placed in the F2 vial with 5 females with brown eyes and wings. We waited 7 days for them to mate. After the F1 adults mated, we were able to clear the F2 vial the same way as before. We followed the same procedure with the fly gnat. Larvae remained in the vial. After the F2 flies had hatched 7 days later, we scored them as well. We again recorded their sex and phenotypes. And here we have the fruit fly life cycle, which shows that it's a little bit over 7 days until they hatch. Time for the results. The initial parental cross between the 7A males and the 6C or sepia females yielded 10 sepia males, 1A male, and 8 sepia females. These results can be seen in the table here. In the second cross, the 5 F1 sepia males were crossed with 5 F1 sepia females. This cross produced 8 sepia males and 22 sepia females. These results can be seen in this table showing the F2 progeny. It appears that the sepia traits for both wings and eye color are dominant autosomal traits. There was only one fly that was not sepia in these crosses, which means it was likely that the A males were recessive for both traits, while the female parents were dominant. There was no real gender bias observed, which suggests that neither trait was sex-linked. If the female parents were homozygous dominant for both traits, and the males were homozygous recessive for both traits, all the offspring would have been heterozygous. This can be seen in the parent-cross Punnett square here. Crossing the heterozygous F1 offspring with each other should have produced 25% recessive offspring, which is here, and those would be those A offspring without wings. We did not necessarily see these numbers, which could be due to the fact that the female flies may not have been virgins when we received them. Female fruit flies can store sperm from previous matings, which could have caused us to see completely different numbers than expected. However, the hypothesis for this experiment proved to be correct, as the sepia traits were in fact dominant and not sex-linked. The lessons learned in this genetics lab prove that working with live animals is not perfect. Things go wrong and flies die, and you have to be able to problem solve. The principles learned in this lab can also apply to humans because it showed how traits can be passed down to offspring. I received some dominant traits from my parents, and that is why I look the way I do, and why I can roll my tongue, just like they can. And here are my references. I so hope you enjoyed this e-report.